Hi, this is Miss Litton, and this is my Period 2 Honors Bio class. Say hi.
Hi.
And we are discussing Chapter 23, Patterns of Inheritance, and this is Part 2. Josh, we miss you today. We think you're weak. You should have been here. Sickness is a weakness. All right. Okay, so for review, I asked you to cross for height and pea plants a heterozygote tall with a homozygous recessive. How would I write that using letters and alleles? How would you represent a heterozygote? Tell.
Big T,
Big T, little t. Perfect. So I said big T, little t. That's a heterozygote. Hetero meaning different. Cross with, how would I write homozygous recessive?
T, little t.
Little t, little t. So then you set up a Punnett square, something like this. Big T, little t, little t, little t. I'm guessing you did this. Now, does it matter where you put your T-ness? No. So you could have put it on either. Sorry. You could have put it on either side. I just tend to. You know, when you use a different letter, that's really bad. I just happen to work from left to right, and that's why I put that there and that there. So when you wrote out your genotype and your phenotype ratios, okay, what do you always do when you write a ratio? Starts with an S. Always simplify. I heard it. Okay. Always simplify. Did I hear it?
No, you heard.
Oh, damn it. So how many different genotypes are represented here? Two. Danger. So how would I write, if there's two different genotypes, what's the first one I would write? One what?
One big little
Big
t.
T, little t. One little t, little t. I simplify it. There's only two different genotypes. And you don't need to write 2 to 2. You always simplify. That would be 1 to 1. Okay. Phenotype, how would I write that? How many different phenotypes are there? One tall to one short. Don't have any naked ratios, no naked numbers. Okay, make sure to always describe it. Tell your bio buddy, if the Punnett square was 1, 2, 3 points, and this was your 4th point, and this was your 5th point, what would you have earned if that was a quiz? Go ahead, tell your bio buddy.
I didn't simplify, so I would have done it. I put it in right.
Okay, so we'll be better on it from here on out. Now, when we ended class last time, we were looking to see if we could taste that PTC paper, right? And I'm going to come back to that in a minute, okay? But some of you definitely could taste it. We're going to just lead up with this review question. I know we went and did one more question farther, but you should be getting 100 on this one. Okay. Guys, so I don't forget, did you see what I posted in Google Classroom about your licorice? Yes. So when does that need to be into that new folder? By Friday, if you're going to do an upgrade. If you have 15 out of 15, do you need to do it over? If you have 14 or 13 out of 15, you don't have to do it. It's only if you want to. Mikey. Natalie, what is going on? And last but not least, when is the review? Do we have a review scheduled? Tomorrow. Oh, no, that's AP, I think that's it. I don't know what it is. You have a review scheduled, I think, for tomorrow, if you want more help with the genetics. Yay! Okay, so you have one. Yeah. A novinator. Okay. There. I better.
I'm scared.
Oh. no, I'm not gonna scroll. I'm not.
We need to banish this person.
It's brutal.
Alright guys, don't fail this one. We're on the board.
Somebody get Mikey a hug. Okay.
I swear.
Sorry means more.
Oh, there we go. We couldn't.
Could have been at 3. I'm just saying.
Just
I'm just saying. Could have been at 3 right now.
Yes.
Is yes the right answer?
What do you think the right answer is?
Yes. Yeah, but what if
You should. This shouldn't count. But I'll let it.
someone's gonna say no.
it should
You can go back to 0 again. Totally your choice.
Okay, wait.
Sorry, I didn't think it'd show up.
Okay, here we go. Now, stop and focus. Okay? Independent assortment, okay? That's only going to happen in meiosis. Only going to happen in meiosis, not in mitosis. In independent assortment, that's when the homologous pairs are in the middle. What stage? Metaphase 1. Now, they can line up, okay? They can line up independently of each other, any which way they want to line up. That is independent assortment. What else do you, look, it says right here, metaphase 1. What else do you see of evidence in that picture? Crossing over, which adds variety. Okay, so this, I'm going to give you an example where this becomes important. Okay, so when we look at this, the genotype of this individual, I'm not just looking at one trait, I am looking at multiple traits now, 2. I don't know if you call that multiple, but 2 traits. Write the genotype of this individual on your practice problems paper. Write the genotype of this individual. How would you write it out? How would you write out their genotype? Don't say it out loud. Write it. When you're done, double tap, and when you're both you and your bio buddy are done, share it with each other. How would you write the genotype for this individual? What stage are they in right now, by the way? Metaphase 1. Exactly. Thank you. Okay, you ready? Did you write that?
Yes.
Okay. Now, let's talk about why it came up that way. We know these are sister chromatids, right? Okay, wait, stop just for 1 minute. Most of you are doing fine, but listen to me. There's a few of you that are talking too much, and what ends up happening is people around you get distracted, and they can't hear me as well, but they're just so polite, they're not calling you out on it. Okay, just stay focused up here when I'm explaining. It gives, and I know some of you are like, I totally get this, I don't need any more help, but others or just don't want to say anything that they don't understand it. So let me explain it every time, and don't bother your bio buddy. Okay, now, when you look up here, these are sister chromatids. That's why I have 2 copies of that little b and 2 copies of that big B. But as far as what the genotype is, I have 1 big B and I have 1 little b. I've just done replication, and that's why you see, so you would not write it out like that. You wouldn't write it like this, right? You wouldn't write that. That's Mutant Ninja Turtle, okay? They just happen to have 2 copies at this time because those are going to end up separating, right? So I have 1 big B, 1 little b, 1 little e, and 1 big E. And you just usually write the dominant one before the recessive one. You keep the B's together and you keep the E's together. What do the B's represent? A homologous pair, right? So I have 2 homologous pairs of chromosomes that I'm looking at. B is on 1, E is on another. What we're going to talk about right now only works if they're on 2 different chromosomes. It doesn't work if they're on the same chromosome. We'll talk about that, and those are called linked genes. Okay, so now, if they line up in this fashion, okay, with that type of genotype, if they line up like that, then when they do anaphase 1, these 2 are going to go be and the first cell from telophase 1, and these 2 will go be in a cell, and then the sister chromatids will separate. So if they line up like this, I'll get a little b, and I'll get a what? Big E. And from this half, I will get a what? Big B and a little e. You all right with that? Okay, but what if, due to independent assortment, if they hadn't lined up like that, what if instead they had lined up like this? Then what kind of gametes could I have? I could have a little b and a little e, and I could have a big B and a big E. Hey, do you see? So depending on how they line up, I have 4 possible gametes. They line up where this way and this way, I have one type, and if it's this way and this way, I have a different type. Now, to figure that out, do you remember FOIL from math? First, outside, inside, and last. So I could take big B, big E, little e, and I could say, first, I'm going to get a big B and a big E. There it is. Outside, I'm going to get a big B and a little e. There it is. Inside, little b, big E. There it is. Last, little b, little e. Okay, now, you do it for this genotype. Write out all the possible gametes for that genotype. Okay, how many different gametes will I get? 4. Tell them to me in order. Go.
Big
Big A, big E.
Big A, little b. Little a, big B. And little a, little b.
Agree? Check with your bio buddy. Shadowlands or sunshine? Shadowlands or sunshine? Okay, questions? Shh.
Will it always be in alphabetical order?
Okay, what you want to do is, regardless of how they present it here, whatever order they give it to you originally, maintain that same order.
Okay.
However they present it. Okay? Now, I want you to do another one for me. And again, do it first by yourself, double tap on your partner's desk, and then share. On the count of 3, show me how many different gametes you have. 1, 2, 3. 2? Looks good. 2. Good. So tell me what they are.
Big T,
okay, wait, don't rush or jump or shout. Here we go. Big T, little R,
little t, little r.
Good on that?
Yep, we're good.
Okay, now, how do you know if you're looking at a gamete, when you're looking at a set of letters, a gamete versus a genotype? Discuss with your bio buddy. How would you know if you're looking at a gamete versus a genotype? Discuss.
This is like the
A genotype or like a genotype is like the different ones together. Gamete.
get the T, big T from.
Okay, so what is this right here that I've circled? A gamete or a genotype?
Genotype.
It's a gamete, yes. Why is it a gamete? I only have one type of T. I only have one type of R. I don't have the homologous pair. So it's a gamete. Whereas this is a what? Genotype. Because I have the homologous pair. Okay, I have it's a genotype, it's my alleles, because I have both of the ones I have received from both my mother and father represented in this genotype. I have my big T and I have my little t, whatever it would be. I have my big R and my little r. If you only see one of it, for instance, if you see this, if somebody gives you this and they say, this right here, is that a gamete or is it a genotype? What would you say?
Gamete.
It has to be a gamete because you only have one Q, one R, and one S. Okay? Write down for me if that's the gamete. There are multiple genotypes that could have come from, but write down 2 different genotypes that the parent could have been in order to generate that. 2 different genotypes. Okay? If you had a gamete, okay, that was QRS, and that is in a sperm, what kind of parent could that come from? Give me 2 different options. There are many options that you could have come from. Give me just 2 different options on the parent. And when you've got those 2 written down, double tap. Now, could the parent have been this?
Yes.
Yeah? Yeah. Could the parent have been this?
That's good. Yeah.
Right? The parent could have been multiple different things as long as each time you add a little q and a little r and a little s in that parent. Yeah? Check with Bio Buddy. Shadowlands or sunshine? Come up with one more possibility for the parent. Come up with a different possibility. I'm not gonna, well, okay, give me one. See if I can do it. Big Q, little q.
Little r,
Little r, little r, little s, little s.
Little r, little r, little s, little s. Would that have worked?
Yeah.
Yes, as long as I have a little q, a little r, and a little s. Okay, if I have 2 of each, then it is their genotype. It is a person. It is a thing. Okay, it has 2 copies of each trait. If it only has 1, then it has to be a gamete. Now, throughout your chapter, you have, I think, about 6 sets of practice problems embedded within the chapter. The answers to all of those practice problems are at the back of the book. Okay. Also, at the end of the chapter, you have multiple choice and more practice problems with the answers at the back of the book. Okay? I don't assign them to you, because I don't like busy work. Because all you would have to do is go to the back of the book and write it up. The key is you should do them for what? Practice, for review. Because your quiz, your tests are always going to be problems, genetics problems that you need to do. So practice doing those genetics problems. Okay? All right. So here is one of them right here. And this is the first practice problem set. I just took a screenshot with my phone on what it looked like in your book. And I just want to do the first 2 on both so that we make sure you understand. But I don't want you to say all the answers to all of them because I want to have everybody have an opportunity to think it through so they can, practice. So let's just do the first one here. It says, for each of the following genotypes, give all possible what? Gametes. So here's a genotype. Do this one right here on your paper. If that is the genotype, in fact, just do the first two here. Do A and B on your paper right now so you can see if you can do it, and we'll check your answer. All right, so give me the first one. All the possible, what are the possible gametes for big W, big W?
Big W.
Big W, big W? Or just what? Just big W. Yeah. If both of them are big W, and big W, every single time you're giving a big W. So you just write it once. Done. Okay, you wouldn't write it twice, and you definitely wouldn't put them together. Why wouldn't you put them together? Because when you make gametes, what law says that they will not go together? Close. Segregation. Law of segregation says big W will be separated from big W. Right? Now, this one, big W, big S, little s, how many possibilities do we have? What are they?
Big W, big S. Big W, little s.
Okay. What law says that you could have big W with big S or big W with little s? Exactly. Law of independent assortment. Don't eat during class. What law says that they are white and they are simple? I'm just making something up there. What law says that they are white and simple? There are only three laws.
Not the segregation one.
Law of dominance.
That's what I said.
See, I just picked the letters. Okay? So, law of dominance says you're going to have that dominant trait, that white over whatever little w would be. Okay? So, law of segregation says your w's are going to be separated from each other and your s's are going to be separated from each other. Law of independent assortment says big W with big S or big W with little s. What would be the exception to that rule? When wouldn't they do independent assortment? When would the big W, let's say, go travel with the big S potentially? If they were on the same chromosome. Then they would have to travel together if they're on the same chromosome. Okay, so back it up to here. Back it up to here. Here we go. Okay, if big W is here and big S is down here, then yeah, law of independent assortment could take place and they could be in any combination. But if, but if big W and big S are on the same chromosome, they will travel together. What's the only thing that would break them up? Crossing over. Okay, crossing over. Okay, I would, not it. One of you is explaining one of the laws, one of you is explaining two of the laws. There are three.
I
Go.
call one of the laws. So, arranged in any way, and it's not affected by other.
Law of dominance is big difference law of segregation.
All right, come back to me. Come back to me. For each of the following, state whether it's a genotype or a gamete. So what would this be right here? Why do you say that? Only one D. What about this one right here? That is a big L, little l. Genotype, because you have a big L and a little l. Want to keep going on these two?
Yeah.
Okay, what would this be?
Gamete.
Why?
Because there's only one P.
One P and one kind of W. What about this one? Genotype. You good? Shadowlands or sunshine? Sunshine? Want to do these three right now?
Yeah. Yes.
Okay, so on your scratch paper, I'll make it bigger. It's asking you to be lame. try again. Go. Okay, so for each of those three, give all the possible gametes for each of those three. Don't say it out loud. Just do it. When you're done, double tap, check with BioBuddy. All right, ready? So what would be, how many different gametes do you have here for C?
2.
What are they? Big T, and then you would put a comma, so I know you're talking about Anna, little t. Okay, 2. Any questions on that one? Okay, how about D? How many different gametes for D? 2. What are they?
Big T, little g, little t, little g.
What law says that this can't be one of the gametes? What law says that you can't have that? Law of segregation. What law says that this individual, let's say, is tall?
Law of dominance.
Okay, good. How many different gametes here?
4.
4. What are they?
Big A, big B. Big A, little b.
Little
Little A, big B.
little
And little
b.
A, little b.
Okay, ask your bio buddy. Shadowlands or sunshine? Clarifying questions with me? Please. Okay, look right here. If I asked you to do that cross and give me genotype and phenotype ratios, could you smell that punnet? Could you see that punnet in your head? You don't even need to do it, right? You can envision it. Okay, envision it, come up with the ratios. You can either write it down or think it in your head, totally up to you. I'll do it up here and we'll see if we matchy-matchy.
Oh. What? You're going too fast for me.
No, I'm just gonna.
Tell your bio buddy, would you have gotten 100% on that one?
Yeah.
A little bit more tight. Clarifying questions? Okay, what about this one? Did we do one like this already?
Yeah, we did.
Something like that, yeah.
Yeah, we did. We're good.
Do it right now. Come up with your genotype, your phenotype. Do it in your head, do it on your practice paper. It might be good to put on your practice paper for review later. You'd be surprised, sometimes people miss things. Widow's peak, the letter is W.
Oh, thank you.
Now, big W's and little W's can look the same, right? So when you write it, like when I do it, I make my big W's like real pointy and I make my little W's very curvy. So I can tell that besides difference in size, so you don't make silly mistakes on a quiz or a test. Things like W's, P's, and S's, because they look the same, uppercase, lowercase, you have to be real careful of. Did you set it up something like this?
Yeah.
Homozygous dominant with a homozygous recessive. So here, if we did genotype and phenotype, there are no ratios. You would just say all what? For genotype, all big W, little w. And for phenotype, you would say all widow's peaks. All right, do this one for me. Shouldn't be anywhere near Okay, did you set it up something like this? Do you know who that is? Jude Law. He has a widow's peak. Okay, so heterozygote with a homozygous recessive. Doesn't matter where you put your W's. No. Did we do one like this at the practice at the very start, just different letters? Yeah, we use height. This should look remarkably, familiar to you. Okay, check with your bio buddy. Shadowland, Sunshine, did they get it right? Did they struggle? Did they need any assistance?
Straight.
Okay. What you are doing with a Punnett square, when you're setting up, you're just doing probability. You're just using a Punnett square to do math, like times tables or something. Okay? So in this case, if I had two coins, and let's say heads represented on one coin a big T, and tails would be the little t, right? So it's when we start to look at more than one trait, we'll do this as well. But right now we're looking at one trait on here. It's like, what if I flip this coin right here? If I flip this coin right here, okay. Oh, thank you. Sorry, I've only done this lesson a million times. I super appreciate that though. And I flip this coin over here, and I have them going. What are the odds that this is going to end up heads? Half of a chance. And the odds that this one's going to end up heads? A half of a chance. So, yes, the independent event, this independent is a half, and this independent is a half, but that they both turn up heads at the same time, then you multiply it together, that's a fourth. Okay, tell me when to stop. Stop. Oh! Totally unplanned. Okay, now, also, I can't, look, you want to do it again?
Stop.
Stop.
But perfect for our next one. Perfect segue. So still, again, a half a chance tails and a half a chance tails were their same, right? They would both be the same, that was a fourth of a chance. That they would both be tails the same, again, what? 1/4 of a chance. Do you see how this is kind of like a Punnett if we were like this? We had 1/4 of a chance that they were tails, and we had 1/4 of a chance that they were heads, right? But now, what are the odds that we would have a heads and a tails? This one is heads, and this specific one heads. This specific one tails.
A quarter.
Still a quarter, right? Guys, listen in. Don't be distracted. If this one in particular were heads and this one in particular were tails, there's a fourth of a chance. For this one to be tails and this one to be heads, again, tails, heads, that would also be a fourth of a chance. And that would be like this one being big T, little t, and this one little t, big T. But it still comes out the same, right? So there's two different ways to get that. So you add them together. You could get it this way or you could get it this way. So really there's a what? 1/2, because you can get a tails, heads, heads, tails. Either way it comes out the same. And that's this right here. Half of a chance, tails, heads. So when you look at an independent event, and then you look at another independent event, that they would turn out that way at the same time, you multiply it together. If there's two ways to get the same thing, that's when you what? Add them together. Okay? That's when you add them together. Youngest bio buddy, take the lead on this right here. Go ahead, explain what was going on in this picture.
So basically we did the place where we said and where so the chance that you're gonna get and then like I think some together so basically we were like trying to get like a specific order
Okay, Shadowlands or Sunshine? Questions? All right, so you have to remember that chance has no memory. So just because if, you know, when you're looking and you're going, a couple has a baby. The first time they have a little girl, what were the odds they would have a little girl? 50%, half of a chance. The next time they go to have another child, ladies, the next time they go to have a child, their little sperm and egg aren't going to go, last time it was a girl, so this time it has to be a boy. Okay? It doesn't work that way. Chance has no memory. The next time it's still going to be half of a chance girl and a half of a chance boy. Chance has no memory. When you talk about happening at the same time in that sequence, you go into labor. And you go into labor and your best friend goes into labor, you know, at that moment, and you have a girl first and she has a girl first, then you can multiply those probabilities together. Because she could have gone first and you could have gone second, and she could have had a boy or you could have a boy. Do you see what I'm saying? That's when you can multiply the probabilities together. Now, another thing when you're looking at these genetics problems is a recessive trait, you know the genotype, right? If somebody has blue eyes, you know their genotype. They are what? Little b, little b. But if somebody has brown eyes, you don't know their genotype. Because they could be, you know their phenotype, it's brown. But because it's a dominant trait, they could be what? Big B, big, what? Big B, big B, or big B, little b. Okay? So when you do a test cross, you're testing an unknown dominant trait because you don't know if it's a homozygote or a heterozygote. If purple flowers are dominant over white flowers, oldest bio buddy, discuss how would the results be different if they were big P or big P little p? How would the Punnett squares turn out differently depending on that purple flower? White is the recessive trait. Discuss, oldest bio buddy.
Alright, so if it was a big P, it would like all over on the spectrum, but if it was here, then it would be 50-50.
But if it's a little p, there is a chance that it could be white. So if it's a brown, it's a 50-50.
All right. So if this was big P, okay, I don't know what he is. I'm testing him with a homozygous recessive. If he's big P, what's this going to be? All purple. But if it was big P, little p, what? Yes. Okay, so now, these are all light-eyed person. Here is my brown-eyed boy. I don't know whether my brown-eyed boy here is big B, or big B, little b. Who does he need to make babies with?
Another brown-eyed person.
Will another brown-eyed person test him?
No.
No. Who does he need to have sex with?
Blue-eyed person.
A blue-eyed person.
Well, the other brown-eyed person could be heterozygote too.
Right, so that's why we can't use that. We have to test him with a homozygous recessive. If he has sex with any one of these girls right here, okay, they all have blue eyes or, you know, light eyes. If he did, okay, that's not, don't do that. Okay. But if he did, and none, if he, like with all of them, okay, and every single one of them had, oh, sorry, he can't have sex with you. If he has sex with all the blue-eyed girls right here, and every one, he has a child with each one of them, and every single child has brown eyes, what do we know probably? He's brown. But if just one of them has a blue-eyed child, what do we know? He was big B, little b. That is a test cross. When you have a dominant trait, you don't know for sure their genotype. Look at me. So you always test them with a homozygous recessive, the recessive trait. That's how you do a test cross.
Is somewhere that paper?
Oh, you want to test somebody?
Well, lot of people.
Well, it's actually
Yes, please.
Focus, people. Good job, Mia. All right.
Thank you.
So if we had someone who could taste PTC paper, and I told you it's a dominant trait. So if they are a taster, that's all is a taster a genotype or a phenotype? Being a taster, is that a genotype or a phenotype? Phenotype. Okay. A taster's genotype could be what?
Big T, little t, or big T, little t.
Yeah, I don't know. This is why I need to test them. So if I had someone who could taste it, a dominant trait, who would we cross them with to see if they were this or this? I would want to cross them with a non-taster. A non-taster that is a phenotype, but there's only one way to be a non-taster. What's the only genotype you can be? Little t, little t. If I make this cross, all of their offspring will be what? Tasters. But if it was this, then about half should be tasters and half should be non-tasters. So I am testing them with a homozygous recessive. Turn towards your bio buddy. Dark-shirted one, explain this question. Go.
So basically, since the tasting is dominant, no matter
So to find out whether they are, it's going to be we're going to eat. So if it's a taster, and there's one child that's not had a big T, little T, because big T, and all tasters.
Wait, how many of you tasted it?
Me and my sister and my dad couldn't, but
Oh, me just okay. All right. For example, your PTC paper should tell you a lot. Okay, go ahead. Tell us. Listen, I'm pulling one of you out as an example. Tell me about your family.
so I couldn't taste the peach.
Okay, wait. So she couldn't taste it. So what would she be?
Too little T.
Too little T. Okay, keep going.
My sister couldn't taste it.
What's her sister?
Little T, little T. Little T, little T. My dad couldn't taste it.
Her dad can taste it, so he is.
Little T, little T.
And your mom? Her mom can taste it. What is her mom's genotype? Tell your bio buddy. So what is her mom's genotype? Do you know?
big T, little t.
Has to be big T, little t. Okay? That's evidence right there of a that would be a good example of a test cross. We know what her second letter is because we know that Dad gave each of his girls a little t. That's all he had to give. Were little t's. Those were his options. Little t or little t. I know. I'll give little t. Mom, however, has the big T option or the little t option because she's a taster. Because they would have to get that second little t from someone. So they would get that from Mom. You all right? Okay. Now, riddle me this. You should be getting 100% here. Did we get 100% on the last question?
It should have been.
It should have.
Wait, what was the context for?
If we cross a taster with a non-taster. If we cross a taster with a non-taster.
They're the same. It's just,
All right, before I hit stop, if we cross a taster with a non-taster, okay, give me the genotype of a non-taster. And all of their offspring, let's see, and all their offspring were tasters. What are tasters?
Big T, big T.
Big T or big T, little t, right? Now, if they are, we know the non-taster is going to give every single time a little t. Every single time it's going to give a little t. So if they're a taster, they have to have a big T. Where did they get that from? Dad. And if every single, we know he's a taster, so he has at least one big T. And if every single time he's giving a big T, then he must be big T, big T. What is that called? Homologous dominant. Almost got it. Almost got it. Good job, Will. No, ¿dónde está? Hang on. Hang on. Is this something you memorize or something you understand? Understand. Understand.
My dad has blue eyes. No, my dad has brown eyes. My mom has blue eyes, and my little brother has blue eyes.
That would make sense. No, okay, so, and if one of their children was a non-taster, then the parent was. It's that unknown parent is who I was referring to. Okay, so they would have to be a heterozygote. Yeah. Okay, now there are more practice problems. We're not doing them right now. Practice problem set 2, you would want to do that, and that would give you really good practice to see if you understood that. Okay? Now, we are going to move on, okay, to get ready for we've done only monohybrid crosses. Now we're going to do a dihybrid cross. Okay? So take a big breath in, let it out. Okay, good. Now, if you had this is one parent, and here is your second parent. Now, here are the traits. Big R is round, little r is wrinkled. Big Y is yellow, little y would be green. So first of all, tell me the possible gametes for this parent and the possible gametes for this parent. Write that out. Possible gametes. We're not doing it out loud because we don't want to take a thinking opportunity away from our bio buddy. Double tap it. And if you think you know the answer to what the possible gametes are, tell me all the different possibilities for the children, what their genotype would be. If you know the gametes, make the children. If you know the gametes, make the children. All right, so I'm just going to randomly, though it doesn't, peas can cross, remember they can cross-fertilize each other, but I'm just going to make this a boy and this a girl, okay? So if this were a boy, what are the possible gametes here? I need more specific.
Big R.
Are there any other possibilities for him? No. Okay? And what are the possibilities here?
Little R.
Little r, little y.
So if this were like us and this were sperm and egg and this sperm fertilized this egg, what are your only options you could have?
Big R.
Big R.
Do you agree with that? Okay. Anybody clarifying questions on that? Okay, now here's step 2. Because they're plants, it doesn't matter. What if I wanted to cross them with each other now. I'm not asking you to do the cross. All I want to know is, since they're exactly the same, right? Just for one of them, tell me all the possible gametes right here. Write it down. What are all the possible gametes? How many possible gametes did you come up with?
4.
4. Were they this? Was that right?
Yeah.
First, outside, inside, last? Yes?
Yeah.
Now, how about that Punnett square?
Oh, how about that?
Oh my.
Oh.
Okay, so if you did a Punnett square on this, you don't need to, okay? I'm going to show you the long way so you understand the thinking, and then I'm going to show you a shortcut, okay? What I want you right now is to conceptualize with me on this, so just be with me, okay? So we would have big R, big Y, big R, little R, big Y, little r, little y. Do you agree with that?
Yes.
Same thing for the other one, yes?
Yes.
Did I set it up right?
Yes.
Why couldn't the gametes be big R, big Y, little r, y? Why couldn't that be something like that?
Segregation.
Law of what?
Segregation.
Segregation. And these are on 2 different chromosomes. That's why I can have a big R with a big Y or a big R with a little y. What law is that? Independent assortment. Okay, now I'm going to ask you to visualize here in a minute, but first I'm going to guide you, but then I'm going to ask you to visualize. If I had this sperm fertilize this egg, it would be big R, big Y, big Y. Do you agree?
Yes.
Big R, big Y, big Y. Do you agree?
Yes.
How would we describe that? As being what? Round. But yes, you would do that too. Round. I want a phenotype. Round and yellow. Yes? Round and yellow. Now, don't get ahead. But just think in your mind first. What if this same sperm fertilized this egg? Think in your head. Now tell me the phenotype out loud.
Round yellow.
Okay, now what prediction could you make based on just this one's gametes?
They're all going to be.
Yeah, do we even care what this one has to say? No, because we know every single time it's going to be round and yellow because it's big R, big Y. There's no debate on that. Is there another row we can do that to?
Yes.
What?
That one.
First column.
First column, because this one is round and yellow, yeah?
Yeah.
Okay, I'm doing it.
Nice.
Now, let's take a look at our second column. Think, what is this one going to be?
Round and green.
And why did you say that?
Big R and Y.
Yeah, exactly. Big R, and little y, yes? So it's round and green. Here.
Round and yellow.
Too much.
Round and green.
Round and green.
So smart. Ask your bio buddy, are you in the Shadowlands or Sunshine? Okay, what am I here? Round and yellow.
What?
Right.
Wrinkled.
And wrinkled and yellow. Little r, big Y, yeah? Wrinkled and yellow. What? Oh, okay. Sorry. Here we go. What's this one? Right here. Wrinkled and yellow.
Round and green. Wrinkled and yellow.
Wrinkled and yellow.
Wrinkled and green.
Okay, this is your task on your practice sheet. Identify all the different phenotypes. Give me a ratio, most dominant to least. Not genotypes, phenotypes. How many different phenotypes are there? Give me the ratio. No nakeds. And when you give me the ratio, give them in the same sequence as I presented them to you in the genotypes, right? Double check with your bio buddy, see if you came up with the same ratio. First of all, let me ask you, how many different phenotypes are there? 4, because I have them here. Okay. All right. What's the first one you would give me? 9.
Brown, yellow, peas.
Okay. Okay then, Papa.
There you
Oh,
go.
and there we go. 9 round, okay. And oh, super annoying. Let's try something else. Okay. Let's go up here. No? Okay.
Oh, look what we got.
Look, now it's writing. Now 9 round and what? Yellow. Okay, give me the next one.
3
Round and green. Give me another one.
3 wrinkled yellow. 1 wrinkled green.
Okay. Now, 9, listen to me. 9 to 3 to 3 to 1 is that's the standard ratio when you do a dihybrid cross. 9 dom, 3 dom recessive, 3 recessive dom, and 1 recessive. Do you see that?
Got it.
What if I had you do a trihybrid cross?
No.
Okay, so our Punnett square keeps getting bigger and bigger. So here's your shortcut. Ready? Watch. When you have a cross like this, here is your shortcut. Watch, wait. You do independent Punnets. So just cross your R's and just cross your Y's all by themselves. Big R, big R, big R, little r, big R, little r, little r, little r, big Y, big Y, big Y, little y, big Y, little y, little y, little y. Now, how many of them would have turned out dominant, round, and yellow? Okay, so this one's round, this one's round, this one's round. How many?
3.
3 out of how many? 4. How many would have turned out yellow?
3.
Yellow, yellow, yellow. 3 out of 4. Let's do some probability. 3 times 3 is 9. 4 times 4 is 16. See how we did that? Dom.
Got it.
Conversely, remember the one little wrinkled green one? How many times would it turn out wrinkled? 1/4. How many times green? 1/4. 1/4 times 1/4. 1 times 1 is 1. 4 times 4 is 16. I don't have to do a big Punnett. In fact, sometimes you will see the Punnets in your head, and then you can just calculate it out. So let's try it. Riddle me this. And give your answer as a fraction on using your clicker. Give your answer as a fraction using your clicker. And I'm going to pause this for a minute because this will take a bit. Okay, so some of you are still finishing this up. Okay? Some of you are still finishing up. Make sure to give your fraction answer. If you're struggling, I'm just going to start you. Hopefully you did it neat, not like I did. So this part, that's an easy punnet because you've done this one a million times. Okay, the next one, big B, little b, big B, big B. You could even make your Punnett square smaller if you wanted to, because these are both big B's up here. And then your last one, the R's. Okay. So the question was, what are the odds of this? So big A, little a, what are the odds of a big A, little a? What are they? 1/2. 1/2. What are the odds of a big B, little b? 1/2. 1/2, right? What are the odds of a big R, little r? 1/2. What is 1 times 1 times 1? 1. 2 times 2 is? 4 times 2 is? 8. Did you get it right?
No.
¿Dónde está?
I put it in before I started doing it.
Oh, spinning wheel of death.
I hate that thing.
Okay, why that is spinning wheel of, oh, cockapoo. Why that is spinning wheel of death.
I hate that thing so much.
Riddle me this one. Don't say anything out loud. Just do it on your paper as we finish up. What are the odds? Just do it on your paper. Don't say anything out loud. What are the odds of getting an offspring who is homozygous recessive for everything? Same parents, homozygous recessive for everything. Don't say anything out loud. If these are the parents, all I'm asking is, if these are the parents, what are the odds of getting homozygous recessive for everything? Don't say anything out loud. These calculations should be fairly quick. Don't say anything out loud. Stop doing math. What are the odds?
0.
Why?
There's no little b.
There's no way to give a little b. There's a way to give everything else, but there's no, so your math should have been 0 odds. Okay? Now, what should you go do? Practice problems. There's another set just with dihybrid crosses in your book, so go do those. And then we'll finish up this chapter next class. Be good. Make smart choices.
For our quiz.